Hello guys, welcome back to my channel. This one's about the rotatory model for ATP generation, basically explaining how the ATP is generated, what are the conformational changes taking place. Before that, let's quickly do chemiosmotic hypothesis because there are many hypotheses that are given out for the mechanism of oxidative phosphorylation. One was chemical coupling, but it was not successful. But then we had the chemiosmotic hypothesis, which we believe now is what happens until further discoveries. So I already talked about it in a video. If you want to know about it in detail, you can go and watch that. But in this, just gonna quickly revise. Chemiosmotic hypothesis just talks about how the transfer of electrons through the ETS is utilized to produce ATP. This is done by the ATP synthase. Normally, in the ETS, what happens is the inner membrane of the mitochondria is impermeable to H plus O plus. So in the ETS, the electron carriers, they actually act as proton pumps. That means they use energy to pump protons to the intermembrane space of the mitochondria, and that's how they create a proton gradient with more H plus in the intermembrane space and less H plus in the matrix of the mitochondria. So that accumulation causes the proton gradient. So throughout the ETS, we see that the transport of electron across the inner mitochondrial membrane is coupled with the transfer of protons. So that's why this is also called as coupling. And the inner membrane, mitochondrial membrane, is called the coupling membrane. So related to this, later we'll see a clinical relation. The chemicals called as uncouplers also. So this is important to know that this thing is called coupling. So the ATP synthase basically allows the diffusion of H plus down the electrochemical gradient, that is through the F naught, that causes the conformational change and eventually the synthesis of ATP, and that's what we're going to discuss. To explain that, Paul Boyer gave the rotatory model for ATP generation. So as I did in the ATP synthase video, talked about the structure of ATP synthase. F1 has 9 polypeptides, 3 alpha, 3 beta, 1 gamma, 1 sigma and 1 epsilon. So here we can only see gamma, the alpha 3 and the beta 3 units, right? Alpha units just bind ATP and ADP, but the actual units that are taking part in the synthesis of ATP are the beta units. So we're gonna see how this whole thing works now. So whenever there is proton flux or when proton passes, that makes the gamma unit physically rotate. It actually physically rotates. So this induces conformational changes in the beta 3 units. Now how is that? Just for explanatory basis, for you to understand, I'm giving this, but I don't back this as the actual thing that happens. Is that if you see here, gamma could be something like this, with the protrusion and with some really specific shapes. So when it rotates, for example, it rotates like that anti-clockwise, the protrusion from here is coming here. That's why this was the tense state first, and now this becomes a tense state. This was in the open state first, now this becomes in the open state. So that's how the rotation of gamma unit is changing the conformation of beta unit. This could be one of the possibilities. But in my MBBS textbook, gamma unit is just shown normally, whereas the shapes are actually given on the outer side of these units. So that's why I don't want you to get serious about this explanation. Moving ahead, we have 3 subunits, O, L and T. No 2 beta units can exist in the same conformation simultaneously. That is an important point. Talking about the 3 states, open, loose and tense. In the open state, the only thing that happens is ATP is released out. There's no binding of any substrate or product. In the L state, ADP and PI exist, but they just exist. There's loose binding. Because of the loose binding, nothing happens in between ADP and PI. Then we have the tense state. Tense state means there's tight binding. That's why ADP and PI form ATP. because of the tight binding. So we see the actual synthesis of ATP is not actually requiring any energy as such, any direct energy as such. It's just the tight binding that it's causing the substrates to bind tightly and form the product. So that is why we say that synthesis of ATP does not require energy. The conformational changes in these states is what requires energy. So coming back to how these changes occur. So the order is loose to tense to open. That's what happens. So when in the loose state is where ADP and PI bind to the beta subunit, that is in the L conformation. So if you see here, the ADP and PI are bound here. So then energy proton flux occurs, there's rotation, and this becomes the tense state. Formation of ATP takes place. I'll come back to this again. These two are actually the same conformations. From here we go to this stage. Tense now becomes open. Now the ATP is released from here, and when the ATP is released, the conformation or the ATP synthase comes back to its original. So as soon as the ATP is released, it comes back to its original conformation. That's what happens every time. So now we follow it from here. So if we see here, this open state is giving its ATP out. Okay, that's what happens in the open state. So for momentarily after ATP release, O is empty. Then conformational change occurs and the loose state is formed where the ADP and PI is actually binding. And then again the same thing happens. It changes into tense. Then it again changes into open. ATP is released. It's empty. Then again it changes into loose and ADP and PI are added. The same thing is happening in the third one also. You have ADP and PI here, tense state ATP, then again open ATP and it is released out. And now it is loose here again. So that's how it happens. That's the whole process of rotatory model or ATP generation. So as I said just now, a few minutes back, that ATP release in O is energy dependent. The actual ATP synthesis is not. The conformational changes, and because of the conformational change only the ATP is being released, right? So that's why that ATP release is energy dependent. So that's very crucial. And then the enzyme ATP synthase acts as a proton driving motor, and is example of rotatory catalysis. That's why it is the world's smallest molecular motor, right? That's that. So this is again explaining the whole process. Protons entering the system cause conformational changes in F1 particle. Initially they are loosely bound to the catalytic site. As the FO accepts protons, the affinity is increased. So that's what they're saying here. They're anyways bound, but the affinity is increased when the protons are passing. So further conformational change induces the catalytic activity and ATP is synthesized. This moves the protons back to the matrix side. That means after the work is done, they go back to the matrix side. Because the whole thing, or the protons are actually going from the intermembrane space to the matrix only. So while they are going only, this whole process is happening. You know what I mean? So after this process is over, like ATP is released, the ATP is synthesized, some more protons are reached to the matrix. As the ATP is released, the original conformation of the enzyme is assumed. As I said, it comes back to its original conformation. The ATP is again bound loosely and the cycle repeats. So that's why the energy surplus that is produced by the proton gradient is stored as chemical energy in the ATP. So that is the rotatory model for ATP generation. That's all for this video. Thanks for watching.